Sir, good afternoon, sir. Yes, sir.
Hello and a warm welcome to everyone. My name is K.O. Satish and I'll be moderating today's session. The topic for today is Shimadzu UFMS solutions for dairy testing. Milk and milk produce is one of the most consumed food items across cultures as it contains all the micronutrients needed for our growth. However, contaminants may be introduced in milk at different points in the dairy chain. Most chemical hazards like mycotoxins, pesticides may be introduced through the feed due to poor agriculture practice or pollution. Over the next few minutes, you'll be taken through a complete workflow solutions and the various regulatory aspects about dairy testing. This includes accurate analysis, the detection levels, and all the solutions that are available to make dairy testing fast, accurate, and simple. At the end of 90 minutes, you'll be exposed to a lot of ideas, methodologies, which you can incorporate in your own analysis, and an opportunity to talk to our experts for any queries you may have. So this webinar is brought to you by Spinco Biotech, India's leading analytical instrumentation company, which was started in 1981. True to its tagline, Spinco is bringing technology and enabling science for the past 41 years. So with over 800 Spincoids and 50 offices across India, we are closer to you than ever before. So it gives me immense pleasure to welcome our panelists for today, Dr. Madasamy, LCMS Specialist and Manager, Spinco Chennai, Dr. Sagar Uttare, Manager, Customer Support Center, Spinco Chennai, Mr. Abhishek Shembekar, Business Development Manager, and Mr. Debajit Sen, Credit Manager. Thank you all for joining, panelists. May I now ask Dr. Madasamy, what can the viewers expect in this webinar? If you can tell us in brief.
Yes, sir. Nowadays, the regulations also stringent related to any food testing, especially for dairy products. For milk and milk testing is huge quantity of market is available in India as well as the international also. For this case, testing laboratories also expecting lot of information related to the dairy testings. Of course, today webinar also we are going to discuss about the dairy testing, which is more useful for food testing industry, sir.
Thank you so much, Madasamy. And also we have Dr. Sagar Uttare, who will be answering all your question and answers. And also he has been the person who has compiled the booklet, and he'll be explaining about the booklet. We also have Mr. Abhishek and DJ. So Mr. Abhishek and DJ, if you can tell us in brief, like what are the topics that you're going to cover towards dairy analysis?
Yeah, so I will be talking about Shimadzu LCMS based solutions for the dairy product testing. How Shimadzu LCMS will be able to help you in achieving load detection limits and to address the challenging and complex metrics like milk or milk products.
Thanks, Abhishek. DJ?
Good afternoon, all our esteemed customers. Today, I will take you through the various parameters which we can analyze through GCMS, GCMSMS, and HPLC technologies of Shimadzu. It may be biological parameters or chemical parameters. So a brief overview of HPLC and GCMS on milk analysis.
Thanks, DJ. So as you heard from the panelists, we have covered all the major topics which gives insights into dairy testing. Also, we have a surprise for you. At the end of today's sessions, we'll be playing Spin the Wheel, and one lucky winner will take home Google Nest. And the rule is very simple. When your name is announced, you have to be present to claim the prize. Else we'll be spinning it once again. So I'd like to welcome Dr. Madasamy, our first speaker, and his title for today is LCMS Solutions for Dairy Testing. Dr. Madasamy has 12 years of experience in food testing and a significant knowledge of ISO 17025, EIC, APIDA, and food safety standards. He has extensively handled instruments like LCMSMS, GCMSMS, HPLC, GC, FTIR, ICP-MS, and AS. Prior to this, he has obtained his PhD from Upasiti Research Institute under Bharatiya University, and he has done his master's in chemistry from Anja College under Madurai Kamaraj University. He has worked with reputed companies like Bureau Veritas Chennai, TV Sud Bangalore, and currently working in Spinco Biotech as a LCMS specialist. Please welcome Dr. MS for his first presentation. So we have some housekeeping rules. Please type in all your queries in the Q&A box only so that we can answer all your queries at the end of this session. So over to you, Dr. Madasamy.
Yes. Thank you, Satish. Yes, is this visible to everyone, sir?
Yes, Dr. Madasamy, can go ahead.
Yeah. Thank you so much, Sadiq. Once again, very good afternoon to everyone. Today, I'm going to discuss about LCMS solution for dairy testing. As I said earlier, it's a very important nowadays because dairy and dairy products grooming in our Indian country as well as the other foreigners also. So in this presentation, I would like to discuss 5 points: dairy market analysis and challenges, typical analysis workflow for dairy sample preparation, mass spectrometry basics, application for dairy analysis, data processing for the reporting. In the year 2020 and 21, India exported the dairy product to other countries almost 54,762 metric tons. These are the 7 states producing huge quantity of milk in India. The dairy market value will be reached almost 30,840 billion in the year of 2027. Because of this reason, India has introduced a new program called National Dairy Program to improve the milk quality. As we know, India is the largest milk producer in the world. The main export products from India is skimmed milk powder, butter, butter oil, ghee and buttermilk. The major export country from India to Turkey, United Arab Emirates, Egypt, Bangladesh, Bhutan, Saudi Arabia, US and Malaysia. There are many highlighted news about the dairy products nowadays on the newspaper. Here, the recent survey had by the FSSAI in India, they found that 7% milk samples found unfit for the consumption. Because of this reason, FSSAI asked industry to reduce the level of unsafe food to less than 1% over next 4 years. From that survey, they said 3.7% of food was found to be unsafe, 15% was non-standard, and 9% was misbranded food in the dairy and dairy products. As per the survey by FSSAI, we can understand that aflatoxin M1 presence in the milk almost 5.7%. Antibiotics presence in 1.2%. Of course, less than 0.1% of pesticides presence in the milk and milk products. There are different regulations followed in domestics as well as the export. In India, we are following Food Safety and Standard Authority of India. When we are going to export the milk from India to other countries, we have to follow EAEC guidelines as well as SFDA for UAE, United States USFDA, Codex for all over India trade. So all these regulations clearly mention that the contaminant should be quantified or quantified through the LCMS or whatever analytical instruments used for this particular analysis. When it comes to the LCMS, we should understand why we have to use LCMS for the contaminant analysis. There are 3 points we have to keep in our mind. First one, scan speed. The purpose of scan speed used to analyze multiple compounds in a single run. For example, nowadays the laboratory is going through a single run, almost covering more than 800 pesticides in a single run, more than 150 antibiotics in a single run. For this case, definitely need higher the scan speed to identify all the data points. As per the regulation, we have to keep 12 to 20 data points in the chromatogram. So if you want to increase the data points, your LCMS should be more than higher the scan speed. Next one, polarity switching. Nowadays we can see many antibiotics having different mode of the nature. It means positive mode and negative mode. This may depends upon the compound nature whether it may taken the proton or released the proton based on the nature. For this case, we have to analyze both positive and negative compounds in a single run. We need very less polarity switching in our LCMS so that we can cover all the compounds in a single run. Third one, sensitivity. Of course, regulation always demanding the LOQ. Based on the LOQ, we have to achieve the lowest direction limit for the particular matrix, especially milk and milk product. We know very well it's a very challenging. For this case, we need higher sensitivity instrument required for your dairy and dairy product analysis. So please keep in your mind when you are going to purchase the LCMS for your laboratory, you can keep these 3 points, main points for your requirement. Currently, the dairy industry facing the challenges. There are 3 challenges. One is sensitivity, because as I said earlier, the regulation always demanding lowest MRL value. For this case, we need higher the sensitivity instrument like ultra fast mass spectrometry for your testing. Also, the ready to method package also available along with the LCMS as a optional to improve your method development without struggling and also you can increase your productivity in your laboratory. The second challenge is sample preparation. We know very well milk and milk product having lot of complex fat molecules. These kind of the fat molecules, we have to use proper sample extraction. For this sample extraction or sample preparation, definitely we need guidelines as well as the application support. The third challenge is data processing. When we are going to discuss about huge data process in a single run, you need the very good software tool that should be user friendly as well as easy data processing to reduce your data processing time at your laboratory. So these are the challenges and solution for your dairy and dairy product analysis in your lab. Let me discuss about typical analysis workflow for dairy sample preparation. There are 3 steps followed in this typical analysis workflow. First one, sample extraction and cleanup. Second one, instrumental analysis. Third one, data processing and reporting. So when we are going to develop the method, we have to maintain 8 steps. First is 7 steps for the method development. Final steps to be covered the validation. So once you complete the validation, you can do it any sample analysis at your lab. So this method development should be focused your target matrix as well as the sample preparation, extraction and cleanup. So you have to optimize all these related parameters which was applicable to your method development should be validated finally before using the sample analysis. There are 3 types of extraction for dairy testing. One is sketches, of course we know very well. Second one, solid phase extraction. Most of the laboratories using this kind of solid phase extraction. Third one, supported liquid extraction. Let me discuss about these 3 types of extraction, how we can improve our sample extraction procedure by using the dairy and dairy products. First one, sketches. We know well, it is a quick, easy, cheap, effective, robust and safe method because the sketches is more environmental friendly compared than other methods. Also, it follows only few simple steps. You can reduce the cost of analysis. Not only this, it will cover all the acidic basic neutral compounds. This is applicable to pesticides, antibiotics. Sometimes it may be applicable to mycotoxins for slight variation changes. The sketches followed in 4 steps. First one, sample weighing. Second, extraction and partitioning. Third one, cleanup. Finally, we are injected into our instrument. Second method is solid phase extraction. This method advantage is it will give clear extract your end of the sample preparation, as well as the higher selectivity for your particular compound and better reproducibility for your matrix. Third one, supported liquid extraction. It will cover all the range of analytes. Not only these, also it will increase the enhancement of ions. The only reason it will collect only the particular compounds to end of your sample analysis preparation. As the last webinar conducted by us, many of the scientists requested to cover the mass spectrometry basics to understand the instrument nature and instrument analytical conditions. So that I would like to discuss about mass spectrometry basics in this presentation. First, we need to understand what is mass spectrometer. Mass spectrometer is nothing but it's an analytical instrument used to measure the ions which was converted from the molecules. It is also called as universal ion detector. From this mass spectrometry, we can get three-dimensional information. First one, retention time information from the LC, molecular fragmentation from the MS, intensity of the molecules also from the LC-MS. We know very well LC in the liquid state, but MS in the high vacuum environmental conditions. How we can connect both different environments? For this case, we have the core technology called interface. The role of interface used to remove the solvent as well as ionize the analytes. There are two types of interface available in the LC-MS. Why we need two types of interface? The compound may be volatile or non-volatile. Of course, the compound is non-volatile. We can go with LC-MS. Depends upon the polarity. Either may we choose ESI or APCI. What is ESI and what is APCI? ESI is nothing but electrospray ionization. Is a soft ionization because the ionization happens in the liquid state. After that evaporated the solvent by the heating gas, it entered to the mass spectrometer. The atmospheric pressure chemical ionization is nothing but the ionization happens in the gaseous state by using the corona discharge needle. So what those are the compounds is not able to analyze in the ESI, we can do it in the APCI, as well as those compounds can able to do it in the GCMS, also able to do it in the APCI. So both these interface can cover all these non-polar to high polar compounds. There are three main parts in our LC-MS. First one, interface. Of course, I mentioned the role of interface to remove the solvent as well as ionize the molecules. Second one, sorting the ions. It means analyzer. Analyzer is nothing but it's a quadrupole. What is quadrupole? Quadrupole is nothing but four metallical rod kept under vacuum conditions from the center of the axis, equidistant from the center of the axis with alternative polarity. This is allow only the particular ions which was mentioned in the software. Finally, detector. Detector will generate the chromatogram from the incident ion. So we have to understand what is multiple reaction monitoring. Multiple reaction monitoring is nothing but it will cover one precursor ion and one product ion. The role of multiple reaction monitoring will increase the sensitivity. Also, it will differentiate triple quad from the single quad. As per the regulation, for example, SANDI guidelines as well as the EU808 guidelines, they record 4 points for this LC-MS. 4 points is nothing but 1 point for the precursor ion, 1.5 point for product ion. So combinedly 2 MRM transition record for your analysis. Also for banned substance, the point is 5. So your point always is covered or retention time also. Of course, the MRF should cover 1 precursor ion, 2 product ion. The total points record is 4 points, almost covered by your LCMSMS. This is the right time to discuss about the application for dairy analysis, because recently implemented regulation by the FSSAI covered many pesticide, antibiotics and aflatoxin M1 also. For this case, definitely I would like to discuss 12 more case studies for this dairy product analysis. The case study 1 related to the antibiotics in milk. We know FSSAI implemented the regulations covered 93 compounds. After that, some reason FSSAI has reduced it to 32 compounds. In these 32 compounds, we have covered 31 compounds in a single run, almost different compound. It means other compounds can be do it with separate method. This method development can be able to do it 1 ppb to 20 ppb covered in this linearity range. Not only this, our team is focusing towards developing method for 93 compounds. Of course, the second case study discuss about 93 compounds. Right now, we are achieving 54 antibiotics analysis in the milk. Not only instrument method, we have covered in sample extraction process also. This method linearity range covered from 1 ppb to 100 ppb. So we have very good method in future as per the FSSAI regulation. Remaining compounds will develop and development is undergoing on. We know the pesticides are also part of the contaminants in milk and milk products. We have very good method for 346 residual pesticides in milk. As per the FSSAI, we have to focus 55 compounds. In this 55, 41 covered by LCMS and 14 covered by GCMS. So in this 41, 2 compounds comes under plant growth regulator, 1 comes under polar compounds. So we need to focus 3 methods for this dairy product analysis. The case study 4 related to vitamin B in infant milk powder. We know vitamin B is essential nutrient needed by the human beings for proper functioning. Vitamin B cannot be produced by human, enough vitamin B cannot be produced by the human. Because of this reason, the vitamin B added from the food sources to the milk powder. So we have very good method for vitamin B in our LCMSMS. We have covered the range from 1 ppb to 500 ppb. Somebody may think that we can analyze in the LC also. Of course, we can do it, but the purpose of LCMS method is to cover very simple sample preparation, also selectivity and sensitivity. Also, you can cover all the compounds in a single run, and you can avoid false positive and false negative identification through your LCMSMS analysis. And vitamin D3 in milk powder is another important one, because we know we can get it through sunlight or through the diet. So vitamin T3 also added in the milk powder. This vitamin T3 method developed based on AOC 2011. We have covered the range 1 to 100 ppb. Of course, this is a very simple extraction process. As I said earlier, importance and essential of this LCMSMS, you can use it. This LCMSMS for this method development. Now we discuss about the protein-rich contaminant called melamine. The melamine contains the rich protein so that it can add it to the raw milk, because the melamine contains 66% of the nitrogen. Also, the diazine diamide also one of the low toxicity contaminants present in the milk, identified in the New Zealand, because it can be used in the agriculture field. The cow may take the cross, it may enter into the milk. For these 2 contaminants, we can develop a single method with very simple extraction procedure. Also, we can covered the range from 0.5 to 10 ppb. So we know from the dilution only we can analyze melamine quickly in our milk as well as the milk powder. So the very good method covered with very lowest limit of quantification as well as limit of detection.
Modules, software.
Before going to next case study, we should think about aflatoxin M1 conversion. Many people think that how it entered to the milk, because aflatoxin B1 is present in the feed taken by the cow. The B1 is converted to aflatoxin M1 in the biological system. So finally, it's entered to the in the milk. So the MRL value for this aflatoxin M1 in domestic 0.5 ppb. For this case study, we have developed a very good method for aflatoxin M1 analysis in LCMSMS, and we have covered 0.05 ppb to 5 ppb. Next one, quantitative analysis of choline in milk powder. You know, choline is also very essential nutrient to our normal function of the body. It can be analyzed in HPLC. The purpose of this method developed in LCMS is not only sensitivity and selectivity. Also, we can use very simple extraction procedure to analyze choline in single quad. So we can cover the range almost 0.5 ppb to 200 ppb of this method development in our milk powder. Comes under the GCMSMS application. We have very good application for MCPD and other 3 compounds like chlorhidyl ester. All different esters we can cover in the GCMS single code as well as the triple code. The purpose of triple code I clearly mentioned earlier. We have to confirm through ion ratio. Also, this kind of ester forming degradation of the fat molecules. So we can use very simple extraction process along with the derivatization process. We can analyze all 3 esters in infant formula. This method developed based on AOAC 2018. The case study 10 related to aflatoxin M1 analysis in milk by HPLC fluorescent detector. Of course, as I said earlier, the aflatoxin M1 can be do it in the LCMSMS. Why? We have to confirm it and we have to achieve the very lowest sensitivity. Those who are focusing European samples, those samples exported from India to European countries, that testing done by using LCMSMS, you can achieve very lowest level. When it comes to HPLC, you can cover 0.1 to 20 ppb along with photochemical reactor. Also, you can use fluorescent detector for this kind of analysis. Next one, 13 bisphenols in milk sample. This bisphenol coating used in milk powder container, they will coated inside the container, and this container should be used for milk powder packing. So this kind of bisphenol can be analyzed in our HPLC fluorescent detector. Also, we can achieve very good R-square value for all 13 bisphenols in milk samples. We have spiked 5 ppb, 100 ppb level. Also, we are getting very good recovery, very good RST for all 13 bisphenols in milk. Final case study discuss about phospholipids, because there are 2 lipids very important in our milk products. So phospholipids analysis in dairy products is very necessary by using analytical instrument. Here I would suggest go with ELST detector for analyzing all these phospholipids in dairy or dairy products. Also, you can get very good sensitivity of this compound through by using this ELST detector. So finally, I would like to discuss about data processing for the reporting. We know that there are 4 steps followed in the data processing workflow. First one, integration. Second one, quantification. Third one, QA/QC. Finally, we are reporting the results. During the integration, we may face that common integration error called, it means proper base-to-base integration error we may get any times. So this kind of the integration we have sort out through special tool in our software. Also, the software should be reduce the data processing time as well as increase the productivity. So it should be highlight the deviation which was identified through more than 800 or 100 comments in a single run, so that you can easily process the data. Not only this, when you are using more than 500, 600 comments, yeah, it should be identify whether it comes under the range or not. Suppose if it is going more than the range, it should show with red color. If it should go less than the range, it should show with blue color. So that kind of identification, that kind of flagging setup necessary for your software usage in your LCMSMS, so that you can improve your productivity in your laboratory. Finally, comes to the summarize my today presentation. Yes, of course, today I have covered the current dairy market status and export trends in India, as well as I have covered the regulations and its challenges and sample extraction and necessity of the cleanup based on the different types of sample extraction procedure. Of course, I covered basics of mass spectrometry and its application necessity in current world for dairy and dairy product analysis. Finally, I said very clearly, data processing workflow depends upon the software, and the software should be user friendly. Thank you so much for your listening. I will complete my presentation. Thank you once again.
Yeah, thank you, Dr. MS. You have extensively covered case studies for various analysis and also given the complete workflow starting from sample extraction, preparation to analysis, and the regulatory aspects required for dairy testing. So we will come back to you for any queries. Before we go to the next part, so we have a poll question here. So I'll just launch the. So for all the viewers here, what is the maximum residue level, MRL, of pesticide residues that is legally tolerated in milk and dairy products according to FSSAI or other regulations? So I shall repeat the question again. What is the maximum residue level of pesticide residue that is legally tolerated in milk and dairy products according to FSSAI or other regulations? So we've got a lot of answers. Well, so I'll just end the poll now in another 5 seconds. 5, 4, 3, 2, 1. Of course, 37% of them believe it's 0.01 milligrams per kg, and 24% believe it's 0.05, and 29% believe it's 0.1. Dr. Madasamy, your comments on this, please.
Yes, sir. Nowadays, we come to know very well about the contaminants related to pesticide as well as antibiotics. Of course, the regulations always demanding lowest MRL value. For these questions, the requirement of this pesticide is almost 0.01 milligram per kilogram in milk and milk products, other food product also. This is the minimum level required for most of the pesticides in food matrix.
Thank you so much, Dr. MS. So this is what it is. So the values are, the MRLs are getting lower and lower. So it needs to update on the regulatory aspects.
Yes.
So Yeah, so we'll now go into the next one. So it's now time to launch our much awaited Shimadzu UFMS solutions for dairy. KMF is the second largest dairy corporation in India with a turnover of 155,000 crores. So they are marketing their products in the brand name of Nandini. KMF were the first to implement all Shimadzu analytical instruments towards their national dairy program for milk quality improvement. So Mr. Muni Reddy, Director KMF, has sent this video towards the launch of the dairy booklet.
Hello everybody, good afternoon. I am Muni Reddy N from Karnataka Milk Federation, working as a Director Quality Assurance. I would like to thank Spinco Biotech for presenting the opportunity for launching the application booklet from Spinco's Shimadzu UFMS solution for dairy.
So Muni Reddy, Director KMF, has sent this video. So now Dr. Sagar Uttare is currently who is working as a Manager Customer Support Center in Spinco Biotech Private Limited. So I'll request him to, who has painstakingly compiled this booklet exclusively for you. And I request him to explain about the booklet. Meanwhile, if you want to get this copy, please send us a mail at info@spincotech.com, or you can even type in at the chat box Spinco, so that we will arrange to send you the copy.
Yeah, thank you, Satish. In fact, we are very happy that Dr. Muni Reddy, from Director QA from Karnataka Milk Federation, has released this booklet. KMF is one of the first dairy cooperatives where Shimadzu LC-MS system was installed, and it is widely used for testing of milk samples. Now we have very good success in dairy segment, and we have almost 7 instruments installed in various dairy industries, apart from other testing labs. So as we all know that India is ranked #1 in terms of milk production, and the industry is expected to grow in coming future. Also, we know that milk is considered as a complete food, but WHO says if it's not safe, it is not food. In this context, milk is contaminated due to the presence of pesticides, antibiotics, various other contaminants. And testing of milk is essential in order to ensure quality and safety of milk. And to have ready methods for testing of milk, Shimadzu has developed various applications for food safety are compiled in this booklet form, which we are now presenting to you. So the booklet contains various applications covering GCMS, MS, LCMS, and HPLC applications for contaminants, various contaminants, including pesticide residues, antibiotic residues, mycotoxin residues, and the quality parameters like vitamins, amino acids, and other aspects. So as you can see, the booklet contains method of determination of 346 pesticide residues in milk using LCMS, MS, and GCMS technology. So this the comprehensive list of 346 pesticides is considered here, considering FSSAI regulation, where around 50-60 pesticides are required to be tested. Again, we have considered European Union regulation, where large number of pesticides need to be monitored in the milk samples. So considering such regulations, a comprehensive list of 346 pesticide residues was considered, and method was developed using LCMS and GCMS. So in this application, you can see sample preparation technique and instrumentation conditions are also given for ready reference. Certain validation data also is included in this booklet.
So Dr. Sagar, if you can make the screen full screen.
Yeah. Yeah. The second application is on veterinary analysis for more than 200 compounds using MRM spectrum mode by LCMS-MS. So Shimadzu has a method package for veterinary drug analysis, and in the package there are multiple MRM transitions listed for antibiotic testing. So with help of all these transitions, the spectrum can be generated in MRM spectrum mode, and the spectrum can be compared with the library, in-house generated library for qualitative screening of the antibiotic residues. So this application is also covered here. The third application is on aminoglycoside antibiotic residues. Aminoglycoside is again one of the class of antibiotic residues, which are high polar in nature, and they are poorly retained on reverse phase chromatography. So we need to use ion pair reagent, but when we use ion pair reagent, they tend to contaminate the instrument frequently. So in this application, helic-based chromatography was developed, avoiding the use of ion pair reagent. And with the help of this technique, all the aminoglycoside residues are analyzed in milk samples, and the data is presented here. Similarly, we have application developed for 45 mycotoxins in baby food samples. We know that aflatoxin M1 only is regulated in terms of milk. However, baby food may contain other products such as rice or flour or other cereal-based products, through which there is possibility of contamination of other mycotoxins. And hence a comprehensive list of 45 mycotoxins was taken into consideration for method development, which covered both positive ionization and negative ionization molecules. And with the help of high polarity switching of 5 milliseconds, this complete analysis is covered in single instrumentation run. So here again, LC conditions and MS conditions are presented, and the validation data is also given. The next application is using Quechers-based sample preparation for aflatoxin analysis. As we all know that for analysis of aflatoxins, we need immunoaffinity cleanup cartridges for having cleaner extract. However, this adds cost to the analysis, and also the method becomes tedious and time consuming. So simple Quechers-based technique is followed here, and with the help of LCMS sensitivity of the instrument, simple technique is followed using Quechers, which avoided use of immunoaffinity cleanup cartridges. Here you can see the sample preparation and LCMS protocol presented here, and the validation data given. The next application is on water-soluble vitamins. Dr. Madhuswamy has covered this in his presentation also. However, we have considered this also in this application, considering the quality aspects of the milk. And the method for analysis of 8 water-soluble vitamins is developed here using an LCMS instrument. The next application is on melamine. So as we know that the limit for a melamine is established by FSSAI as 2.5 ppm in all food matrices. So this is again a simple dilution method, and using LCMS sensitivity of the LCMS, this method can be easily adopted in the laboratories for analysis of melamine in milk powder and milk samples as well. So again, the instrument conditions and sample preparation method is given here for analysis of melamine. We can see amino acids also is one of the quality parameters, and with the help of LCMS technique, we can avoid derivation. Derivation is again a long process, and with the help of LCMS, derivation process is not required, and we can analyze around 20 amino acids in this instrumentation in using LCMS, and the method details are given here. Products in M1 can be analyzed using simple HPLC technique also instead of LCMS, using fluorescence detection, and the same application also is covered in this booklet. We can see again the method for determination of 13 bisphenols, which may be present in sample or which can leach into the milk or milk products through packaging materials. And we have a HPLC based method using fluorescence detection for analysis of all the 13 bisphenols in milk samples. Similarly, we have method for analysis of MCPDs and fatty acid esters using GCMS with the help of derivation, and the method details are given here in this booklet. The last application is on milk sugars, which can be analyzed using again fluorescence detection, and the details of methods are given in this booklet. So we hope that this booklet will be handy and a good reference for the testing laboratories for method development and use of these methods for analysis of milk samples. Thank you.
Thank you, Dr. Sagar, for explaining in detail about the contents of the booklet. I think we have already received overwhelming response for the requirement for the application booklet, and shall arrange to send the same in the next few days. So we also interviewed Mr. Munir Reddy, Director of Quality Assurance, Karnataka Milk Federation, who are currently using our Shimadzu instruments as explained before for dairy analysis. And here are some of the excerpts of the video.
Normally, the dairy products contain some contaminants like pesticides, antibiotics, toxins are present in trace level, very minute level. To identify these trace level contaminants, it is required sophisticated instruments like LCMS, GCMS, and ICMS are very necessarily required. So industry nowadays we have to invest on these analytical instruments to get precisely the contaminants. KMF lab is an accredited lab. So presently with available facilities, we are testing all parameters of antibiotics, pesticides and toxins, vitamins, chemical parameters which are in trace level, which are necessary for fulfilling the quality and safety of the product as per FSC. We are receiving amazing support from Spinco since time of installation and developing application like method development and validation also, and moreover they are extending very good support in maintenance and maintenance of the instrument.
Yeah, so continuing, our next speaker is Mr. Abhishek Shanvekar. So he is currently the business development manager in Spinco for the past 13 years. He started his career as a product specialist and now he is the business development manager. So he is a business manager who always puts customers first. His thorough understanding of the subject makes him your technical solutions provider. So he'll be presenting about Shimadzu LCMS for dairy product testings. Over to you, Abhishek.
So thanks for the introduction. Yeah, so I hope my slide is visible.
One second, not yet.
Hello.
Yeah, it's still not yet.
You can stop share and oh yeah, now
can.
it's visible. Yes. Share it again, Abhishek.
Yes, sir. Is it okay now?
Yes, 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 yes.
Yeah. So hello everyone. I'm Abhishek Shembekar from Spinco Biotech, working as business manager for Shimadzu LCMS product line. Today I will give presentation on Shimadzu LCMS and its key features with the differentiating UFMS technology. Since we are today having this webinar on dairy solutions, dairy product and testing, I would just like to talk a couple of minutes on the white revolution. Government of India initiated Operation Flood, known as the White Revolution, after witnessing the huge success of Green Revolution in India. White Revolution in India started with the motive of increasing milk production to make the country as one of the largest producers of milk in the world. Operation Flood, launched on 13th January 1970, was world's first and largest dairy development program, and it has been a landmark project for National Dairy Development Board, NDDB. It transformed India from a milk deficient nation into world's largest milk producer. Here, I would like to acknowledge Dr. Varghese Kurian, chairman and founder of Amul, who is also known as father of White Revolution in India. So with this, I would just like to start with my presentation on Shimadzu UFMS solutions for dairy product testing. LCMS today is very important, very useful, and a very powerful tool for qualitative and quantitative analysis. Triple quadrupole spectrometry is a versatile tool that can be adapted to a wide range of applications in pharma, CRO, chemicals, and very into the food analysis, the food testing. In terms of instrumentation, researchers today require speed, sensitivity, and flexibility to adapt to new challenges into the industry. For years, Shimadzu has been known as the leader in liquid chromatography products, and now Shimadzu is being recognized as a leading manufacturer of LCMS instrument in the industry. Yeah. So Shimadzu Corporation Japan is the longest serving analytical instrumentation manufacturer in the world. Since 1875, Shimadzu has been introducing various cutting edge technologies and serving the scientific community with their innovations. Spinco Biotech, headquartered at Chennai, they are the distributor to Shimadzu for their entire liquid chromatography mass spectrometry product range. Spinco, we have 50 offices, 50 support centers across the country with a team of close to 800 people to support all our valued customers in terms of application support, service support, or any other technical solutions required. We also have a state-of-art lab at our Chennai facility to support customers in terms of any advanced training, method development support needed. Customization has been the cornerstone of our company culture. Shimadzu is one-stop solution for all your analytical requirements. From UV spectrophotometer to high-end LC-MS systems, all the analytical instruments are manufactured by Shimadzu. From Spinco, we represent Shimadzu for the liquid chromatography and LC-MS product range, the HPLCs, UHPLC, preparative HPLCs, and LC-MS technology. Having said that, innovation has been Shimadzu's DNA. Shimadzu has also highest number of patents in the industry, so which is the highest number of patents by any vendor in the industry. Shimadzu is also blessed to work with a Nobel laureate, Dr. Koichi Tanaka, who won Nobel Prize in 2002 for his work on MALDI. He has been part of the cutting edge technologies and the new innovations which are coming out in the form of new instruments, new technologies by Shimadzu. He has been part of all those innovations. Recently, Shimadzu also completed 50 years of MS innovations. It was 1970 when Shimadzu introduced the first mass spectrometer. Those days it was magnetic sector-based mass spectrometer. And over these years, Shimadzu has been introducing various cutting-edge technologies, various MS technologies, be it GCMS technology, LCMS technology, and the very latest and the recent UFMS technology. Talking about the references and the installation base, today we all know the importance of LCMS into any analytical lab for a low-level quantification to address any complex matrices. LCMS is very much essential, and we have from A to Z companies, Shimadzu LCMS, we have references, be it from from Azidus to Zydus, all the companies are having Shimadzu LCMS. These are spread across various industry segments, pharma, CRO, chemical industries, diagnostic industries, and whatnot, and very mainly the food industry. Talking about the testing labs, a few referral nodal testing labs like QV, Eurofins, Vimta, they have been using multiple Shimadzu LCMS MS systems, and these systems are used for various food commodity analysis. And milk is one of the most challenging and very important commodity for analysis. Similarly, we have seen increase in regulation, FSC and the awareness in the food quality. Food manufacturers also started using, developing and upgrading their in-house labs for the in-house quality check. They have been using LCMS instrument for various parameters and tests. Specifically, if I talk about the references into the dairy industry for milk and milk product analysis, we have been working with dairy and dairy from Punjab, or KMF, the brand name of Nandini, Milma from Kerala, and Saras Dairy, which is the Rajasthan Dairy Federation, and even the private dairies like Hudson Agro, who has a brand of Arun ice creams. So they have been using Shimadzu LCMS for various, the major for them are the FSSA pesticide list and the antibiotics list, which is available. So these are the primary contaminants and parameters which they would be doing it. And it all started with under this milk quality milk program under a national program for dairy. So where in 2019, when Department of Animal Husbandry and Dairy, DAHD, they have issued a notification, and they have implemented this program for upgradation of the labs, where they talk about 4 major contaminants like pesticides, antibiotics, aflatoxin, melamine. So where these instruments like LCMS-MS and GCMS-MS become very essential. Yeah, so the UFMS technology, you have been listening about UFMS, ultra-fast mass spectrometry. So the significance and the need of UFMS technology Basically, UFMS as a technology is mainly built on three important features: the speed, polarity switching, and sensitivity. So, Shimadzu offers world's fastest mass spectrometers with this UFMS technology, all without any compromise in speed. Ultra fast scanning speed, ultra fast polarity switching, and the sensitivity have been the pillars for differentiating UFMS technology. Shimadzu LCMS 8050 is the revolutionary model which is being used by all these dairy cooperatives which I have shared. So this has been a very revolutionary and a very successful model. And these are the few key specifications like scanning speed of 30,000 Daltons per second, ultra fast polarity switching, 5 milliseconds, sensitivity best in class, and ultra fast MRM 555 channels per second. The very unique thing about Shimadzu LCMS technology is the on-site upgradability. All the Shimadzu instruments can be upgraded to a higher sensitivity system on-site. Yeah, so this gives a future-proof investment to all the users. So any model, be it 8045 or 8050, these models can be upgraded in your lab itself at any given point of time in future. Shimadzu LCMS 8050 offers unparalleled measurement speed and high sensitivity performance, redefining LCMS analysis for multi-component analysis and reliable quantitative analysis. It uses the latest N-series UHPLC, which has got analytical intelligence into it. It talks of, so it's a fully automated system. The N-series UHPLC has been designed to meet new customer challenges and requirements. Shimadzu HPLC's outstanding hardware performance and the latest digital technologies such as Internet of Things, machine-to-machine learning brings us to achieve the high reliable data acquisition with improvement of operational efficiency. The N-series UHPLC is a fully automated HPLC system, UHPLC system with auto startup to auto shutdown. It can also complete the auto purging, column equilibration, baseline checks in advance, and be ready for analysis as soon as the user arrives in the lab. Moreover, the system can be set up in advance to run without analysis till the shutdown. So, combining Shimadzu LCMS with the N-series UHPLC, the analytical intelligence functions offer various flexible workflows from instrument preparation, analytical data processing, ensuring high-quality analysis with analytical intelligence functions like smart functions, column-friendly flow palette functions, automatic system diagnostic functions, mobile face monitoring, and much more. Talking about the construction and the technology inside the mass spectrometer Shimadzu 8050, Shimadzu UFMS series of LCMS systems are the linear triple quadrupole design, and it has got an orthogonal design for ion transfer. Yeah, so as in the previous presentation, Dr. Madaswamy was explaining about the internal architecture and the principle of ionization into the basic slide. There are three parts: one, ionization, and the ion transmission and the detection. So when we talk about the ionization source, the interface, basically, there are two types of ionization sources: ESI and APCI, electrospray ionization and atmospheric pressure chemical ionization. So in Shimadzu, we have got heated ESI source, which gives the better desolvation efficiency, which enhances the formation of ions, and then we have an APCI source. So here the point is the newly designed unit, the heated ESI and the APCI can be interchanged without use of any tools. There are no cables, no tubings, hence the changing of these sources becomes very easy and user friendly. Talking about the cleaning of system, the cleaning and maintenance, the ESI capillary or the desolvation line, it can be cleaned without breaking the vacuum. So it is very easy and very quick. A user himself can do the regular cleaning part. You don't have to depend on engineer for the regular cleaning part. So regular cleaning or periodic cleaning is required because food samples are complex. There is a lot of contamination which goes in. So there is a regular cleaning which is required for the system. So every time you don't have to depend on engineer for the cleaning process. And this cleaning can be done without breaking the vacuum. Talking about the ion optics, the UFQ array and ultra-fast lenses, again, these have been the patented design by Shimadzu. With this new design, ions are precisely converged and the signal loss are minimized. They enable smooth ion transmission of all the ions towards the quadrupole rods. In addition, the efficiency of the ionization process is improved with the use of heated ESI source. So here I would like to talk about the significance of UFMS technology. So when we talk about, we all have seen and experienced when HPLCs have been evolved to a UHPLC technology. So when we talk about UHPLC as a front end, we all talk about shorter run times, sharper peak, good resolution. So if you have this kind of data coming up from a UHPLC as a front end into an MS, you should have a fast MS also. To complement a UHPLC, we need a fast MS. So that is where UFMS technology comes into the picture. And when in UHPLC, the peaks are eluted in a fraction of seconds, if acquisition speed or the scanning speed of a mass spectrometer is slow, it will collect less data points. But if a mass spectrometer has got a fast scanning speed or a fast acquisition speed, it will collect sufficient number of data points so that your peak shape is Gaussian and you'll be able to do good quantitation of your experiments. And as we know, for a good shape or for a Gaussian shape, it is a minimum 10 data points, 10 to 15 data points are needed to form a good peak and a good quantitation. Yeah, so this is one example where you can see there are 9 data points and the percentage RSD is 8.75%. Here, this experiment was run with a low scanning speed, but when the same experiment is performed at 30,000 daltons per second speed, the number of data points are increased to 13 and the percentage RSD has been significantly improved to 2.45%. Second, the ultra-fast polarity switching. So this becomes very important because when we talk about pesticides analysis or antibiotic analysis, these are the methods are multi-analyte or multi-component. So when we talk about 100 or 200 pesticides in a single run, we know a few of them will be positively charged, some of them will be negatively charged. So in order to develop a single method without any compromise in sensitivity, an instrument should have a very fast polarity switching. So Shimadzu LCMS system gives a polarity switching time of 5 milliseconds. So both positive and negative ionization can be done simultaneously without any compromise in sensitivity. So another part is the collision cell technology. Again, when we compare it with the conventional collision cell design, as you know that ions travel inside a vacuum. So when they travel inside a vacuum, they pass through quadrupole 1, they come to quadrupole 2, where the fragmentation takes place. So an external gas pressure is applied an argon gas is used for collision purpose. So when the ions get fragmented, they get break, there is a loss of energy. So in this process, the transmission efficiency is affected. So because of that, there will be a degradation in detection sensitivity because the transmission of ions has been affected. There will be a crosstalk or a formation of ghost peaks because you will be running multiple events one after another. And there is a chances of omission of analyte information. So in case of Shimadzu US sweeper collision cell, which is a patented design by Shimadzu, there is a pseudo potential formed, something like a small entry, large exit, where ions travel and cover a longer path. So in spite of having a fragmentation or a loss of the momentum, they cover a longer path and this sweeps out very fast out of collision cell. So because of this, there is no loss in sensitivity and the sensitivity is maintained throughout the system, and the crosstalk or the carryover is almost zero. And because of this, we are able to run multiple MRMs one after another. If I talk about the combination effect of these three important features, the fast scanning speed, fast polarity switching, and zero carryover because of the US sweeper collision cell technologies, we are able to produce this kind of data. You can see this is the practical data which was performed, and 647 pesticides are analyzed in just 10.5 minutes. And as you can see this data, and each compound has got 3 MRM transitions. So this is only possible when instrument is having a fast scanning speed and fast polarity switching. So in our case, Shimadzu instruments provide fastest scanning speed of 30,000 Daltons per second and a fastest polarity switching of 5 milliseconds. So this is a combiningly with UHPLC as a front end and UFMS as a mass spectrometer, it gives you a killer combination. So we all know why UHPLC, because of shorter run times, better sensitivity, a good chromatographic resolution in order to overall lower the operating cost and comparatively, and ultra fast MS for having fast scanning, fast polarity switching, faster data processing, and the ultra high sensitivity. So this in combination provides ultra high sensitivity, a better specificity, a higher throughput. So which is these three things are basic requirements for any analytical laboratory today. This was all about the hardware, the unique features, and how significantly these are important in different contaminant analysis. But at the end, user is going to sit on computer. They have to handle the software. Software should be easy and user friendly. So Shimadzu Lab Solutions LCMS with Insight is a very easy and a user friendly software. It takes scans, all the checkpoints it nicely takes care, and it has got a very interactive view where in a single window you get all the information. The Lab Solutions Insight software, which is for easy data processing, gives all the information in a single window. You can see compound sample information, compound information, chromatogram, linearity, everything in a single window. So this makes the job of user very easy. When you have a lot of data to be analyzed, it can be done in few minutes. There is also a flagging function available where you can color code the samples and define a threshold. So when you do the data review, the samples which are going to pass, which are going to fail, you can color code them. You can define a threshold and software can color code and do the flagging for those. So this, again, the data review mechanism becomes very easy for the user. There are few unique method packages which are supplied along with the instruments as per your need, as per your customized requirement. These method packages or databases can be supplied to you. Shimadzu has a database for residual pesticides. We have a database for mycotoxins, veterinary drugs, antibiotics. There is a GCMS database available. So these method packages or databases are basically they comes with all the method conditions, all the information are stored in that, so that you can quickly download, optimize your instrument, and start the analysis. You don't have to start from zero. So the development time is basically saved. So for example, Shimadzu LCMS database for pesticide residue, it has got 836 compounds registered into the database, and it comes with all the necessary information inside the database. So it can be quickly optimized, and you can start your experiments. Talking about the support and how to run the instrument for a new user or an expert user today, you need support in terms of the development, in terms of training, in terms of handling different kinds of matrices, different challenging food commodities and different contaminants. So we have got a very big support team. These are field application chemists who are available. They help you in terms of the complete hand-holding process, in terms of training, method development support, any kind of support needed. So they are there in place, the field application chemists. And then we have 4 customer support centers across the country. Shimadzu's application lab in Mumbai and Delhi to support our customers in West and the north part of India. These customer support centers from Shimadzu are full-fledged with the complete instruments from Shimadzu. All the instruments, UV to mass spectrometer, everything is available in our Mumbai lab. A team of application chemists are available to train you and again help you in terms of any kind of method development process. And there is a state-of-art lab, as I was telling you, in Chennai. It is Pinco's Customer Support Center. So these labs or customer support centers can be utilized for any advanced training, method development support, or any other kind of support needed to our users. In addition to that, we have got the largest field support team, the engineers basically, which have been the backbone of Spinco. So we have got more than 500 engineers, field engineers today across the country to support and ensure your instrument has zero downtime and the uptime should be maximum. So the largest service support team which is available, and the very innovative cutting edge, which is our technical journal. So it gets published every month. So over these many years, over the last 12 years, we have been circulating every month our monthly publication, cutting edge scientific journal. So 80,000 print digital copies and 12,000 print copies are circulated. It consists of scientific articles, trending applications, application notes, and many other useful scientific information. So you can always subscribe to receive your copy, a monthly copy for this cutting edge journal. I also take this opportunity to invite articles or any scientific articles which you would like to share with scientific community. You can reach us and we can publish anything which is helpful for the scientific community. So again, just to summarize, the Shimadzu LCMS or UFMS technology becomes the instrument of choice. One is the multi-analyte methods. With the help of fast scanning speed and the polarity switching, you can run multi-analyte methods. More number of analytes can be clubbed in a single method. Good chromatographic separation, thanks to UHPLC, N-series UHPLC with the AI technology, analytical intelligence, wider dynamic range, high selectivity to achieve the low LOD, LOQs, and to provide a high throughput to your lab. So with this, I would just like to end with my presentation.
Thank you, Abhishek, for giving us the various options of Shimadzu UFMS technology for a dairy analysis. So before we go to Debajit, so we have a small video which we'd like to play about Shimadzu LCMS.
Shimadzu's ultra-fast mass spectrometry technology is focused on improving three main instrument attributes: acquisition speed, sensitivity, and robust operation. UFMS reduces the waiting time during analysis and also reduces the time wasted on instrument maintenance between testing runs so that our triple quad instruments can dramatically improve productivity for labs. To hit these new benchmarks, Shimadzu engineers developed innovations to three critical systems: the collision cell, the ion optics, and the ion source. At the heart of ultra-fast MRM acquisition is enhanced collision cell technology we call UF Sweeper. With UF Sweeper, we apply voltage to the collision cell to actively sweep away residual ions and speed up the entire acquisition process. UF Sweeper cuts the pause time to 1 millisecond and instantly starts the next round of MRM data acquisition. The result is a total cycle time of 1.8 milliseconds per MRM. This is the shortest cycle time on the market, delivering 555 MRM transitions per second. And this speed is achieved with no compromise in sensitivity. In order to measure MRM transitions in very short times, you need very high sensitivity. So we developed our UF-Q array ion optics technology to enhance LC-MS instrument sensitivity. UF-Q array is an ion guide based on a high-frequency quadrupole electric field with a high central convergence. It improves ion focusing efficiency to achieve higher signal intensity. For this design, we opened up the ion inlet and narrowed the aperture at the end of the array. This converges the ion beam precisely into the quadrupole mass analyzer. The wider gas flow path ensures that inert particles do not interfere with the ion path. Background noise is reduced while signal intensity is increased up to 5 times. This unique approach routinely delivers robust detection at very high sensitivities, even with complex matrices at trace levels. UFMS is about more than just speeding up acquisition and sensitivity. It also provides robust operation. In other instruments, there was always a trade-off between the efficiency of the ion transmission and the amount of contamination brought into the system. But not with Shimadzu triple quads. In our latest models, the ion source is now called the ion focus unit, and we have found the optimal placement, increasing the distance from the probe to the ion inlet ever so slightly. This design retains the high sensitivity you need while bringing in a lot less contamination. So you get the robust performance you need as well. And less contamination means less time wasted on instrument downtime. You can run it longer before needing to stop for routine cleaning and maintenance. Shimadzu UFMS delivers the speed, sensitivity, and robustness that together increase your productivity. Contact your local Shimadzu representative for more technical details and to discuss your specific application.
Excellence in science. Shimadzu.
So our next speaker is Mr. Devajit Sen, who has over 16 years of experience and is currently working with Spinco Biotech for the past 14.5 years. So he is the territory manager for eastern part of India and has a passion for chromatography systems, especially GCMS. He has a good understanding of the GCMS for the past 14 years, and needless to say, he is a self-made expert on mass spectrometry. So he'll be presenting about Shimadzu GCMS and HPLC solutions for dairy. Over to you, DJ.
Thank you, Kevin, sir. Good evening, esteemed customers. It's my pleasure presenting Shimadzu GCMS and HPLC solutions to our customers. Over the next few minutes, I will take you through the hardware part of Shimadzu GCMS solutions as well as HPLC. This is a very easy and simple chart or to understand basically the HPLC and GC applications when it comes to the JD applications. When it comes to the chemical properties, basically GC, GCMS, LCMS, and QTOF, all these instruments can be used for the residual pesticides, diazonamide, melamines, fatty acids, triglycerides, dioxins, veterinary drugs, hormones, steroids, vitamins, and antibiotics. Here you can use as well as GC and HPLC, MS technologies, LCMSMS as well as GCMSMS. When it comes to the biological properties, aflatoxins, mycotoxins, and E. coli, etc., can be analyzed by HPLC and LC-MS, both technologies. As Avishek has already mentioned, Shimadzu has patented UFMS technology, ultra-fast mass spectrometry technologies. Here, the front end of GC is Nexus model 2030. The same model has been used in single quadruple as well as triple quadruple, which is a universal GC today. GCMS 2020NX is the single quadruple model, GCMS, and triple quadruple Shimadzu has two models, 8040 and 8050. We will talk about more 8040, which is suitable and sufficient for your dairy applications. The USP of Shimadzu GCMS technology is smart performance, smart productivity, as well as smart operation. You can see the picture here. The GCMS right side is the Nexus GC2030 and left side is the MS. It's the smallest footprint available in the market, occupies the minimum bench space. The tower in front is the auto sampler with a round carousel of more than 100 sample vials capacity. The GC itself is having a state-of-the-art user interface, graphical user interface. It's a touchscreen display where you can see the real-time chromatogram, the various flow parameters, whether system is ready or not, that also will be indicated in the panel itself. As I mentioned, the Nexus GC is called the universal GC, which has various latest features which no other competition can provide. Basically, this system can use any kind of carrier gases, be it hydrogen, helium, or nitrogen. Hydrogen, as we all know, is an explosive gas. So when it comes to hydrogen, we have to be very, very cautious. So leak sensor is very, very important. Here, Shimadzu Nexus is having a leak sensor inbuilt. So hydrogen leakage, absolutely not an worry. This is the basically the process of starting a GCMS right from beginning to analysis. You have to start the full line pump first, then check the vacuum level for TMP. You can start the TMP, then leak check again. Then stabilization, you have to wait for the tuning and tuning judgment. So this is a general process cycle where in general circumstances, you don't have the clear idea of what is the time required for the various steps. But here in Shimadzu GCMS, we have active time management feature. Here you can see the starting full line pump is the one work which the user needs to do in the morning while coming to the lab. Next, all the parts, all the steps are automated. Then the tuning judgment, tuning is done, and finally you can start the analysis. So at various steps, the time remaining will be showed in active mode. When it comes to the ionization techniques in GCMS, we all know that hard ionization is EI, which is the most common ionization technique available or used for 90% of the applications. But it may happen so that you need some molecular ion information at times. What we do is we use the PCI, the CI technology. Here, if you want to change the ion source from EI to CI or vice versa, the common problem is you need to shut down the vacuum. So EI analysis, you stop vacuum, change the ion source, again start up the vacuum, again you can do the PCI analysis. That's the common phenomena. Whereas Shimadzu is having a smart EI-CI source. The beauty of it is you don't require to stop the vacuum. It can give you EI data as well as you can do CI analysis. Here you can see EI analysis. So you need not stop the vacuum and directly you can switch on to PCI analysis. When it comes to smart performance, yes, the system is having quite a few features which basically enhances the system capability. The first feature or first technology what is used in the ion source itself is a shield plate has been introduced, which reduces the filament potential. And in that case, the ions are directly reaching to the detector in maximum numbers. We are not losing any ion. Sensitivity is enhanced. Second technology what Shimadzu uses is a pre-rod, that is, which is acting as a pre-filter before the quadruple. So it's made up of a different material. So the quadruple main rods are not contaminated. Unlike some of the companies which need to use quadruple heating, which is not good for the quadruple. So we don't require any heating for removing the contamination in the quadruple because the quadruples are not contaminated. Basically, there is a pre-filter in terms of pre-rod. Then Shimadzu uses off-axis ion optics. The reason for using this kind of off-axis design is basically to remove the neutrals from entering to the mass spectrometer and create noises. UFMS technology is a patented technology of Shimadzu, which allows 800 transitions per second, which is the highest available in the market. So there is no crosstalk absolutely in the collision cell. The next technology is called ASSP, Advanced Scan Sim Protocol. This enables a high-speed scanning of 20,000 mass unit per second, again, which is highest in the market. And then you don't lose any data point in between. So stable data intensity you can get with high-speed MRM. This is how the ASSP works. Generally, the ion travels like this from the source to the quadruple, so multiple collisions, and then it takes time. But here, when ASSP is on, it can move very fast and then improves the sensitivity. When it comes to maintenance, Shimadzu has changed a lot from the previous models, as well as it is not available with any other company in the market. We are using split tech technology, which is basically a easy to fill kind of a nut, which can be easily rotated without the use of a spanner or any kind of a tool. So the inlet maintenance or the column changing, everything is very, very easy. Split filters are also transparent filters. So you understand whenever the requirement of changing the filter comes, you can easily change it. Absolutely no tool required for the maintenance of injection port or the split filter. Shimadzu has revolutionized the change this technology of sensitivity increase by adapting to 2030 technology of AFC, advanced flow controller. It's a digitized flow controller of the next generation, which increases the sensitivity to many folds. Coming to the turbomolecular pump, which we can say as the heart of a GCMS or any kind of a MS system, because we need vacuum for the ions to travel from source to quadruple and quadruple to detector. So the entire path should be under vacuum. More the vacuum, more is the ions uninterrupted flow. So we get more sensitivity. Here, the technology is dual differential pump technology design. Basically, one part of the turbomolecular pump is kept below the ion source. The other part is kept below the quadruple. So in that case, what we achieve is a uniform vacuum throughout the chamber, which ensures highest vacuum and then highest sensitivity as well. Then Shimadzu is having this twin line GC. That is, there are two inlets connected to two columns, and simultaneously operation can be done, two injections can be made. So when it comes to labs productivity increase, all these technologies play major roles: active time management, Shimadzu smart database, click tech technology, and smart EICI ion source. This is the smart pesticide database available with Shimadzu with more than 530 MRM transitions, and then compound information with retention indices and cache numbers. Transition information is also available. It can monitor ions, collision energies, ion ratios. MRM optimization tool is also available, which is very, very user friendly to do the analysis. Irrespective of whether the person or the user is novice or experienced, he can use the machine very easily. Self-optimized product ions and collision energies are set up. Lab Solutions Inside is a beautiful post-run analysis tool which is incorporated by Shimadzu, introduced by Shimadzu, and it is very, very popular in the scientific fraternity. It uses color flagging options. You can set your threshold for the data, what you can accept. Below the threshold is accepted. At threshold, it can be required to be double checked. It can be in a different color like yellow, and above threshold, it can be green, where the data processing becomes very easy. So it may be 3 days job, can be minimized to 1 day job or 1 hour job. This is a post-run analysis tool. Quick DV is another option for available with Shimadzu. In conventional method, what happens? You require to prepare the test sample, then the standard sample preparation, and then you can do check the analytical conditions. All these will take up one day, and the second day you can do the instrumental analysis, create the calibration start, and then calibration curve, then qualitative analysis. All together it may happen. So 3 days are occupied. Here, quick DB of Shimadzu, you just prepare a test sample and then add surrogates. Then what you can do is directly start analyzing with analytical conditions which are already optimized in your quick DB. These are various examples or protocols of milk or milk products. We are having a lot of methods developed already. GCMS and GCMS, 98 pesticides as per FSSI, multi-residue analysis, determination of melamine. So we have a lot of data created. Simultaneous determination of 19 phthalate compounds in milk powder, the GCMS. Now we are moving to HPLC. Hardware a part of HPLC. Shimadzu has already completed 25,000 number of HPLC installations in India, which is a remarkable feat. When it comes to the HPLC requirement in the market, there are basically 3 categories of HPLCs. First is 3 to 5 micron requirement particle size analysis, where all-round P-series HPLC is available with Shimadzu. Next is a mid-level UHPLC, which caters to the requirement of 2.5 micron particle size columns. The solution from Shimadzu side is advanced I-series instrument. The third category is the real ultra-fast HPLC with sub 2 micron category, where we have the next generation UHPLC called N-series. P-series is our modular system. It is called proven performance with productivity, what this system can offer. It's a complete modular system. So all modules are box type. It's a towering in design. Pump is having the lowest pulsation. Autosampler is highest throughput autosampler. We have a choice of many detectors, UV, RID, PDF, fluorescence, or you can add MS also to this. We have choice of column ovens of multiple columns. P-series has a new feature called ASP, automated system preparation. What this feature is, I will explain. Basically, whenever a person comes in the lab in the morning, you have certain protocol to start the machine. You have to start the system, then check the system, check is required, auto purging, column equilibrium, you have to check the baseline, system suitability test, then you can start your sample analysis, and finally you can shut down. Here all these steps, right from system startup to shutdown, everything is automated till the analysis. So basically it's called automated system preparation, ASP. This, once you click this ASP, after a few minutes you can see the green light glowing called system is ready. So in between the person can do any other work. So it's direct productivity increase of a lab. Fluorescence detector of Shimadzu is available in the market, which is having the highest sensitivity of 2000:1. Fluorescence can be used along with the Pickering lab UV post-column reactor or post-column derivatization unit, which is a AOAC accepted methodology, which can give you the highest sensitivity when it comes to the mycotoxin or the aflatoxin analysis. This is a beautiful solution available. We have a lot of papers generated. ELSD is another detector which is universal detector, and then it is becoming popular day by day. Many labs are using. We have this ELSD which goes to the nanogram level of detection, and it gives 10 times higher sensitivity than UV or fluorescence. The next mid-level UHPLC, or we call it advanced I-series UHPLC. It's basically living LC, and Shimadzu is the first company in the world which introduced the word analytical intelligence and living LC. So basically, what is living LC and analytical intelligence? Living technology is an emerging wave of innovation that incorporates principles, activities, and dynamics of life in technology. Basically, today we are surrounded by many new latest technologies of this living analytical, this artificial intelligence like self-driving or transportation, AI-powered robotics, surgery, and many other things. So Shimadzu talks about 2050. So model name is 2050. Basically, we are futuristic. Whatever technologies people are thinking today, Shimadzu has already brought to you. So autonomous technology, this can be used in terms of S2S, IoT, MPM. So startup to shutdown, Internet of Things, mobile phase monitoring. All these latest features are incorporated with this analytical intelligence. Basically, no matter if the user is very novice, so he can be treated as expert. The operations that system enables and treats itself so that it is operated by an expert. So it automatically monitors and diagnoses itself as per that. It has auto diagnostics and recovery functions based on AI. Shimadzu is also having another latest technology through which it is called ICM, basically interactive communication mode. Today, everybody is using smart devices like smartphone, iPhone, tabs, etc. So whenever the person is inside the campus, somewhere outside the lab, he can still monitor the system in a smart device with a software called in an app format, Lab Solution Direct. So you can directly monitor the system, see the chromatogram, flow rate, etc. It's real-time monitoring of the system. Then there is some more technologies which is called Work from Anywhere Kit, WFA Kit. In terms of COVID and post-COVID, we are all habituated to new normal, which is working from home or working from anywhere, different locations, remote locations. So this technology enables the user with some additional power. What is that? If your mobile phase is running short, automatically the user will get a notification in his device that mobile phaser is running out. So before you start the batch itself, with a gravimetric sensor inbuilt in the tray, will automatically get notified about this mobile phase running out or insufficiency. So you can give instruction to the person available in the lab and automatically this job can be done. N series is Shimadzu's latest development in UHPLC segment. Basically, this system is built on 42 patented features and it got a prestigious award called Red Dot Design Award. So it is uninterrupted operation, fully automated S2S, starting up to shut down. As Abhishek has already informed about this system, so every step is automated. Mobile phase monitoring and then gives alert, auto diagnostics, flow pilot, and then management utility. Flow pilot, I will explain, it's also a patented feature. Basically, all labs are concerned about the consumables cost. And then your column life is very, very important. What happens? General phenomena, left side graph, if you can look at, generally the column oven requires some time to stabilize its temperature. Whatever temperature you set, it will take some time to get stabilized to that temperature. Till that, your flow rate is increasing, means your pressure is also suddenly increasing on the column. So in order to overcome that, your column life automatically gets deteriorated. In that case, what happens is, this is a patented technology of Shimadzu called flow pilot. Your flow will get increased gradually. Whatever flow rate you are setting, for example, 1 ml per minute, it takes exactly half of this flow, 0.5, till the time unless the column oven temperature is achieved to set temperature. With this gradual increase in temperature and set flow rate, you are saving your column, safeguarding your column, and longevity of the column becomes much more. Then there is a AI scouting analysis available with Shimadzu. Multiple columns and multiple solvents you can switch over, and then you can do method development with these. And there is a software also, method scouting software, which can provide to the customers. So it is very easy for method development, which takes generally 3 to 4 days time. It will take only 1 to 2 hours. New HPLC N series is a very high throughput system. It takes only 6.7 injection cycle time. That means injection to injection is only 6.7 seconds, which is the lowest time available in the market. And it has the highest throughput auto sampler with rack changer facility of you can do 17,000 samples also. It has ultra lowest carryover, 0.0003. Nothing is detected actually. And it has many latest functions like reagent addition, dilution, co-injection, online dissolution, overlap injections, stack injections, mixing, derivatization. So system is equipped and built in with lot of innovative features which are very new to our customers and they will really enjoy it. So we welcome you to the future of HPLC and to the future of GCMS and GCMS-MS. Thank you for listening patiently.
Thank you, DJ, for giving the insights on GCMS techniques for dairy applications. Yeah, so we now go into the question and answer sessions. I think there are quite a few questions. I'll just quick start with one of the questions to Dr. Sagar. So Dr. Sagar, so you are heading the customer support center in Chennai. So what is the kind of support that is being provided from the customer support center?
In Chennai, we have a customer support center where we have half a dozen mask model to 8610X, our high-end model. So we do develop methods on these instruments, and we support customers for training. We support customers for doing method developments and transfer of those methods to customer site.
Right. So, and does it also include the complete validation of methods, or is it just a method transfer that has been supported?
We do a partial validation of the method. We don't do complete validation, but we do transfer the method, and then our field application specialist will support customers in doing method development or method transfer, and then further validation of the method.
Okay. And since the regulations are becoming stringent, and LOQs are also getting lower, so suppose a method has been developed today at certain levels, and then FSSAI demands a much lower level. So what does the customer need to do at that time? Do you need to validate the complete method once again, or?
If the method is established with certain LOQs which are meeting the current requirements, in that case, the method needs some modification either. With the method modification, if we are able to achieve the new MRL requirements, then the method needs validation again. Or else we have to go with the high sensitivity of the instrument, and then the same method can be adopted, and then further validation can be performed new instrument.
Right. So this question is to Dr. Madaswamy. So you were emphasizing more on speed of the instrument and how the systems are having high scanning speed and lower switching speed. So for a user, like what does it mean for a user who is using a high speed system?
For example, if you are going to analyze any compounds in the LCMS, we need a very good sensitivity as well as the higher scan speed instrument, so that we can cover all the data points. We can measure the different sides of the compound nature through our LCMS. For this case, definitely I suggested 3 important points: sensitivity, scan speed, and polarity switching. Because depends upon the physical and chemical nature, we have to adopt all these compounds to our high sensitivity LCMS. So that is very, very important when you are going to choosing the LCMS.
Right. And so this question is to Abhishek. Like, since there are too many options available, so we also have different models of LCMS available to cater to the current requirement and also to the future requirement. So what are the things that one user needs to consider before buying a system?
Yeah, so basically what we recommend is to users is to go with their requirement rather to instead of going with the highest sensitivity system available, because that will have its own challenges when you bring it to the actual usage. So that is where we recommend users to go with what they need specifically. And that is where Shimadzu also introduced the concept of on-site upgradation, which is part of our technology. So starting from 8045 platform, the Shimadzu LCMS 8045 model today is able to address and meet all the domestic as well as regulatory requirements in terms of the MRLs or LOQ limits.
Right. And when it comes,
even after 5 years, the levels are going down. Yeah. So when the levels are going down, so even in that case, instead of investing on a new machine, users have a provision to upgrade the existing system. So that is again a very unique differentiating thing about Shimadzu MS technology.
Thanks, Abhishek. And so this is again to Dr. Madaswamy. And so when considering the instrument LCMS, so what are the software, probably the softwares and the method packages that needs to be considered for dairy applications?
When you are going with LCMS analysis, we have to go with the user-friendly software that should be easily handled by the scientist in the laboratory. Also, why I have to mention the method package, it should be very helpful to the scientist who is working with the LCMS analysis. For example, if they want to develop 800 compounds in a single run, the optimization or manual optimization takes more and more time. Instead of this, if you're having the method package, just you can copy the environment transition, whatever you have related to your method, you can copy and paste it just to verify the method through your standards. Also, this methods package should be covered all your LC parameter as well as the mass parameter. For example, RT, gradient program, everything is mentioned in this method package. Definitely, it would be support for your testing laboratory for dairy and dairy product analysis as well as other food matrix also.
Yeah, and to Dr. Sagar, like suppose a customer buys a new mass spectrometer and he's got significantly a new user who was just using HPLC. So how soon will it be possible for them to adapt to a mass spec, or what is the kind of support that you will be providing to the customer or to the user to adapt to the MS technology for lower analysis?
Yeah, we do have a very good field application support team. In fact, we have around 50 field application people who are spread across the country, and they are supporting customers. They do handholding of the customers for getting like to know about the instrument initially, and again after that they support customers to develop methods on their instrument, and then further after a few days we again provide like advanced training also to the users. So this way we do complete handholding of the customer from starting from installation of the instrument to development of method and then validation of the methods. And again, in terms of achieving lower sensitivity or those kind of things, we do support them for method development at their customer side, or we also do method development and CSE, and those methods will be transferred to customer.
Yeah, thanks again. So this question is for DJ. Since you were talking about GCMS and the GCMS-MS, so what is the preferred technique? So do a customer needs to consider GCMS-MS or a GCMS for dairy analysis?
GCMS-MS is always preferred over GCMS. What happens in case of a closely associated fix or the coeluted fix, GCMS is having further resolving power, which in case of a detection also, you can go to trace level. So GCMS-MS is a more preferred technique.
Thanks. And so you were also mentioning about the different HPLCs. One is the P series and the AI series and the N series. I understand N series is the UHPLC system. So as an user, what should they consider? Should they consider a P series as a front-end system, or a AI series or an N series before actually buying a mass spectrometer?
Yeah, basically in my first slide, it was explained with respect to the market demand and requirement. It goes into three categories: it's a 3 to 5 micron analysis, and then 2.5 micron, and then sub 2 micron category. So if it is ultra fast analysis requirement for the customers and wants to use a sub 2 micron column, in that case, N series UHPLs will be recommended. For general purpose analytical usage, 5 micron based P series is sufficient. And for mid-level HPLC with lot of innovative technologies, we recommend AI series. It also depends on this kind of categorical analysis and also the budget requirements.
Sure.
2 factors.
Yeah, and so currently, if a customer is using only HPLC for aflatoxin analysis, so what is the recommended model? So should they go with the post-column derivatization with the fluorescence detector, or should we suggest a ELST detector for this analysis?
Yeah, fluorescence detector with post-column reactor will be the ideal combination. As mentioned, it's also AOSC approved technology and gives very high sensitive analysis results. And Shimadzu fluorescence detector is having the highest sensitivity in the world. So yes, customers can go with this combination.
And for the Abhishek, so apart from the HPLC and the MS, so what are the things that they need to consider while buying the system? So what are is there anything that they need to consider for the sample preparation or any other things that is additionally required apart from the LCMS?
Yeah, basically a lot of sample preparation instruments are needed before actually taking the sample for LCMS or GCMS analysis. So there are basic lab instruments like the sample solvent filtration unit, sonicator, centrifuge, water purification system, balance. So all these are instruments which are, there are a list of instruments which are available, and these are needed basically for a sample preparation for injecting into an MS analysis.
Right. And Dr. Sagar, do you suggest quetchers to be, as a sample preparation to be chosen, or they can do a manual sample preparation?
Quetchers is like a very simple technique, and it is effective, and it can be easily adopted in the laboratories. Whereas earlier days methods were specific to group specific, we can say, or compound specific methods which were very tedious, and they were also prone to errors since many steps were involved into those sample preparation techniques. So since quetchers is very simple, errors are also less, and it is environment friendly also. So for certain applications like pesticides or even some antibiotics, we recommend to use quetchers based technique.
And in the dairy testing, so where are the points? So like, do they have to do the analysis while checking for the raw materials, or on the finished products, or what are the stages of testing that has been done?
As of now, as part of safety or quality program, the industries can do testing at like source also, or even at collection points. Whereas in terms of food safety regulatory requirements or meeting the regulatory requirements, it can be on finished products also.
So we got quite a few questions. There's one question from Kaligana Durga Prasad. So it says, which approved standard method needs to be followed for the validation process?
For validation, for pesticide residues, it is generally SANTI is one of the validation protocol which is followed. Whereas for antibiotics, as far as EU countries are concerned, they have EC 808 regulation, which is released recently, and that is upgraded version, we can say, of earlier EC 657 guidelines. So these two are generally followed validation guidelines. Apart from that, for mycotoxins, it is 401 of EU regulation. So there are certain regulatory or say validation guidelines available which are adopted. Or in general, AOSC method validation criteria also can be followed.
Thanks. So there's another question from Mr. Keshavan Nambudri. So if formalin is added in milk, whether it will affect the actual value of aflatoxin M1?
Yes, shall I answer?
Yes, go ahead.
Yes. Yes, actually, if you are adding, people are doing very smart work in their industry. Sometimes aflatoxin is very high. They are planning to add the formalin. That is wrong option. But even though if you are adding formalin, definitely it will affect aflatoxin M1 quantification. Suppose the milk contains aflatoxin M1 in 1 ppm. If you are adding formalin, it will comes down to 0.25 ppm or less than that. Definitely it will affect aflatoxin M1 if you are adding the formalin into the milk.
So there's another question from an anonymous attendee who asks, which regulations needs to be approved for testing dairy products?
Yes, there was many regulations available based on their import and export, but they will not decide. It's not only the particular. For example, India, we have to focus FSSAI guideline. It means they will guide the test to do like this way, like that. They will guide, but they will not. It means they will not specify the particular product and matrix. They will mention that this is the compound and this is the MRR value for the particular matrix. That's all. They will provide the regulations based on their committee decision.
Yeah, so there's another question from Chetan. Is it mandatory to add water during pesticide extraction while analyzing skimmed milk powder?
Yes, we need to add water because milk powder contains the dry one. So whenever we are taking dry powder or water maybe, we have to definitely add water so that the water can used to dissolve the material what we are added in the during the process extraction process. Definitely, I would suggest you to add water whenever you are going with a dry matrix. For example, raw milk also better to add less 50% amount. It means instead of 10 ml, you may add 5 ml to get good recovery of your compounds.
Yes. So going to the next question, I am using an UHPLC with PD and RF detector. So how many pesticides can be analyzed with this instrument?
Of course, PDA, for HPLC with PDA, you can analyze because PDA having multiple element, it means array. So you can add 20 compounds also you can able to do it, but not in the PPP level. Only possible in the ppm level. It means 1 ppm or 0.5 ppm. Definitely it would not fulfill the regulation guidelines. So you can go with LCMS is the better solution for your pesticide residue analysis.
Going on to the next question, have you developed a combined method for antibiotics, tetracyclines, fluoroquinols, sulfonamides combined in milk?
Yes, we have developed very good method for this kind of all group of compounds like sulfonamides, quinolans and tetracycline, including tetracycline UFMS. The challenges is here, we have to separate both parent and UFMS so that we can achieve through the instrument method. When we are going to analyze the milk matrix, we are a little bit struggle to develop the matrix. It means sample preparation, we are struggling to separate this compound. Also, we are struggling to get the sensitivity of the particular UFMS. So for this case, we are working now to develop the extraction procedure as well. So currently, we are having very good method for the instrument. We are future, we are working together for this combined method.
So there's one other question. Is sample digester required in LCMS for pesticides residue testing? Sample digester required.
Actually, digestion is not required for pesticide residue analysis, only required for heavy metals analysis.
Okay. So yeah, I think we have come to the end of the questions. There's one question, probably SRM can answer. What will be the cost for the upgradation of equipment?
Yeah, I think this is from Dr. G.K. Naik, right?
Yes.
Yeah, I have already requested his contact details. I will talk to him and understand what is the current HPLC model, what he has, and then propose the upgradation suitably.
Sure.
Yeah, I think we have to share his contact details so that we will discuss with him and propose the suitable upgradation proposal.
Sure. So I think we have answered all the question and answers. I think it was a more comprehensive webinar and most of the things were answered in the presentation. So now we have come to the most exciting part where we will be announcing the winner of the Google Nest. And I now request Mahesh to take over and do the spinning the wheel.
Yeah, hi. Thank you, sir. And thank you everyone for being online until the end of the presentation and the webinar. I hope the session is informative and engaging. So we have finally arrived. Time has come for us to spin the wheel, and if I can have the wheel on the screen at this point of time.
1 second.
So I would like to explain how this is going to work. So we have a a spinning a wheel of fortune, I would say, available with this, where we have all the participants who has started when the webinar was started. All the participants who have logged in this session are available in this one. So once you spin the wheel, it will pop out a random, completely random name. So all you have to do is you need to type in your name. Once you see your name on the screen, you need to type in your name. And if you're comfortable, maybe we can ask you to come online and you can have a word with the panelists, and who wins is yours. So can we have the spin?
Yes. So I'm just clicking.
Yeah. So it's a completely random one. So we don't know who is the lucky one. So let's see who is going to be the winner. It's coming down most. Okay, almost. Yeah, I think that's how the luck works. So Mr. Arun Mishra, if you're available, I request you to please post it in the chat, your name in the chat, so that we know that you're online. Mr. Arun Mishra. Oh, I don't see him online available. Can we have a spin once again?
Yeah.
So he has missed an opportunity.
Yeah. Finally. Rucha Paddeshi. Do we have Miss Rucha Paddeshi? Okay, we don't have them available at this point of time. We need to go once again. Let's see.
So we have only 3 rounds, right?
Yeah.
Yeah. So next one is the last.
Yeah.
If they are present, they are going to get it, or this will be postponed to the next webinar.
RJ Meenakshi, sorry. So do we have Jay Meenakshi? No, I don't see them available. I don't see them online.
Probably we can have one more last round.
Yeah. Nitin Sharma. Nitin Sharma. I don't see Mr. Nitin Sharma available again. Over to you, sir.
Yeah. Okay, so we'll randomly pick a winner and then hand it over to them. So thank you so much for joining our webinar and your overwhelming response. So we have received all your requests for the application booklet and we'll ensure to send the same to you in a couple of days. And please do follow our LinkedIn page so you all get the updates of our upcoming webinar. Once again, from Team Spinco, thank you for attending this webinar and have a wonderful weekend. Thank you so much.
Thank you.